In this section, we are going to discuss estimation of risk and rules of autosomal dominant inheritance. Estimation of risk for offspring with autosomal dominant inheritance. In first case, heterozygote paired with a normal homozygote. These were the results. So, out of 4, 2 were heterozygote, that were diseased, and 2 were normal homozygotes. Risk is 2 by 4, or 50%. In second case, heterozygote paired with another heterozygote. So, out of 4 offsprings, 1 was dominant homozygous, that was diseased. 2 were heterozygote, they were also diseased. And 1 was normal homozygote. 3 were diseased, out of 4. So, the risk is 3 by 4, or 75%. In third case, dominant homozygote paired with a normal homozygote. All offsprings were heterozygote, that were diseased. 4 out of 4 offsprings were diseased. So, risk is 4 by 4, or 100%. Now let's discuss the rules for autosomal dominant inheritance. There are the basic rules for simple autosomal dominant inheritance. These rules apply only to conditions of complete penetrance, and where no novel mutation has arisen. First rule is that both males and females express the allele, and can transmit it equally to sons and daughters. Let's understand this rule though pedigree. Male who is heterozygote and is diseased, and female who is normal homozygote. Let's discuss the next generation. Normal homozygote male, diseased heterozygote female, diseased heterozygote female, and normal homozygote male. If normal homozygote male paired with diseased heterozygote female, let's discuss the next generation. Normal homozygote male, normal homozygote female, normal homozygote male, normal homozygote male, diseased heterozygote male, diseased heterozygote female, diseased heterozygote female. If diseased heterozygote female with normal homozygote male, diseased heterozygote female, diseased heterozygote male, diseased heterozygote male, normal homozygote female, normal homozygote male, and normal homozygote male. So, in this pedigree, 3 males and 3 females are affected. So, both males and females express the allele, and can transmit it equally to sons and daughters. Moving towards second rule for autosomal dominant inheritance. Every affected person has an affected parent. As in pedigree, you can see this is affected parent. His both daughters are affected. His third generation is also affected. Moving towards third rule for autosomal dominant inheritance, and that is, in affected families, the ratio of affected to unaffected children is almost always 1:1. Let's understand it. The first condition in humans for which the mode of inheritance was elucidated was brachydactyly, characterized by abnormally short phalanges. In Mendelian symbols, dominant allele B causes brachydactyly, and every affected individual is either a homozygote or a heterozygote. In practice, most are heterozygotes because brachydactyly is a rarer trait. So, heterozygote paired with a normal homozygote. These are gametes generated from father, and these are gametes generated from mother, capital B from father, and small b from mother. The offspring is heterozygote. So the offspring is diseased. Small b from father, and small b from mother. So the offspring is normal homozygote. Capital B from father, and small b from mother. The offspring is heterozygote. So the offspring is diseased. Small b from father, and small b from mother. So the offspring is normal homozygote. So 2 offspring are diseased, and 2 are normal. So 2:2, 2 one's the 2, and 2 one's the 2. So we can say, in affected families, the ratio of affected to unaffected children is almost always 1:1. Moving towards estimation of mutation rate. The frequency of dominant diseases in families with no prior cases can be used to estimate the natural frequency of new point mutations. This varies widely between genes, but averages about 1 mutational event in any specific gene per 500,000 zygotes. Almost all point mutations arise in sperm, each containing, at the latest estimates, 20 to 25,000 genes. There are therefore perhaps 25,000 mutations per 500,000 sperm. So we can expect around 5% of viable sperm and babies to carry a new genetic mutation. However, only a minority of these occurs within genes that produce clinically significant effects or would behave as dominant traits. So, it was all about this section of the video on autosomal inheritance and pharmacogenetics. In our upcoming section, we are going to discuss pharmacogenetics. But have you solved the question I've asked from you in our previous section? Well, you will get the answer in the end of this video.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.